Hi, I'm Joshua Egalicia, and I'm about to discuss to you photosynthesis. Have you ever wondered how plants eat? To answer this question, you need to understand photosynthesis. Photosynthesis is a process which plants make their own food to grow and develop. In order to form photosynthesis, they need various elements such as sunlight, carbon dioxide, and water. There are two stages of photosynthesis: light-dependent reaction, which uses sunlight, and light-independent reaction, or Calvin cycle. Now, we're going to the light-dependent reaction. First step in the electron transport chain is photoexcitation. We need a photon to excite an electron. The energy from the photon makes it go from a high to lower energy state, allowing it to power reactions. Photoexcitation occurs at two complexes, photosystem 2 and 1. Each of these photosystems is full of chlorophyll molecules. When a photon hits one of the chlorophyll molecules in the photosystem 2, resonance energy causes the energy from that photon to hop from the one chlorophyll molecule to the next within the complex until it reaches the chlorophyll in the reaction center. Here, an electron is excited in its high energy state. It has potential to do work for the cell. This process occurs twice, producing two excited electrons. Meanwhile, the oxygen evolving complex, which is converted to an unstable state by the incoming electron, is busy splitting water molecules. This serves two purposes. Firstly, let the electron used by a photosystem 2 are replenished by splitting to one water molecule as a byproduct. 1 2 water molecules have been split, the oxygen atoms combine in 2O2. The plant considers this a waste product. The second purpose of the water splitting involves two hydrogens releases into the lumen. These protons serve to increase the proton gradient so that there is a higher concentration of protons in the lumen. Then in the stroma, the greater the gradient, the more badly the protons will want to diffuse to the other side. Next, QB picks up two excited electrons from the reaction center, chlorophylls, in photosystem 2, along with two protons from the stroma and shuttles them to cytochrome B6F. Here the plastokinobi passes the electron to the complex and releases to the two photons into the lumen cytochrome B6F, then adds a further two protons into the lumen. Further building up the gradient, plastocyanine now picks up two electrons and transfers them to photosystem 1. The electron receive an additional energy boost with the help of two additional photons and get transferred into the last mobile carrier, pyridoxine. Pyridoxine then transport the electrons to the pyridoxine NADP reductase, which uses the two electron plus a photon from the stroma to convert NADP plus to the higher energy molecule NADPH. Note that this also contributes to the photon gradient by removing a photon from the stroma. So now we come to ATP synthase. Now we're going to light-independent reaction, or Calvin cycle. Now we come to the light-independent reaction, in other words the Calvin cycle. Remember, the purpose of the cycle is to use the high-energy molecules created in the light-dependent reaction to produce useful carbon-based molecules such as glucose, cut off RuBp and carbon dioxide. The plant absorbed from the surrounding air by its stomata. The first step of this cycle, in which carbon dioxide is attached to RuBp, is catalyzed by the enzyme ribosco. This process is termed carbon fixation, since carbon is being converted from a gaseous form as CO2 into a solid. Now the addition of carbon dioxide to RuBp created an unstable 6-carbon intermediate quickly breaks down into the 2,3 carbon chains. These 2,3 carbon chains are called PGAs. Next, a coupling of reaction occurs. This is how cells use ATP, other high-energy molecules, to do work. After charging, these molecules have decoupled a catabolic exothermic backwards reactions to endothermic reaction. In this case, the energy released from the breakdown of two ATP molecules is used to convert two unstable NADPH molecules back into NADP+. With the protons from the NADPH is being added to PGAs to form two G3Ps, which are the ultimate product of the Calvin cycle. One of these G3Ps can leave the cycle and can be used to build glucose, a 6-carbon molecule, and other carbohydrates to add to the plant's biomass. The other G3P now uses ATP to get converted back to RUBP, so the cycle can repeat. But hang on, something doesn't quite add up. We produce two G3Ps, each of which has 3 carbon. How are we going to produce glucose, which has 6 carbons, and RUBP, which has 5 carbons? In reality, we have to consider 6 rounds of the Calvin cycle, all happening at the same time. We actually need 6 CO2 molecules to enter the Calvin cycle to produce 1 molecule of glucose, one of each carbon. With these 6 cycles happening at once, we have 12 G3Ps produced for a total of 36 carbons.